Now, structural organization of eukaryotic cell. We all know that cells shape, size varies, but the structural organization in every cell is almost same. In structural organization of eukaryotic cell, there are three fundamental regions: plasma membrane, cytoplasm, nucleus. What is plasma membrane here? It is also called the plasma lemma or cell membrane. Nature of this plasma membrane is thin, flexible, living, transparent, and semi-permeable in nature. Semi-permeable is here selective transportation of material. Now, structure. Most accepted structure of plasma membrane is fluid mosaic model. You can see in the diagram. This is the fluid mosaic model of plasma membrane. It is made up of phospholipid bilayer, protein molecules, carbohydrates molecule, and the phospholipid is present in bilayer, which has head and tail. Head is hydrophilic or tail is hydrophobic. Hydrophilic means water loving and hydrophobic means water heating. One large size molecule of protein is present in the center, that is known as the tunnel protein molecule. This is the fluid mosaic model of plasma membrane. Now, what are the functions of plasma membrane? It regulates the exchange of material between the cytoplasm and extracellular fluid inside the cell. It also allows the selective movement of materials because these are semi-permeable membranes. Now, mechanism involved in semi-permeability. Mechanisms are divided into two: passive processes and active processes. In passive processes, there are two processes: diffusion and osmosis. So, firstly, what is diffusion? Diffusion, it is the spontaneous movement of material from a region of its higher concentration to region of lower concentration. For example, if we take water in a beaker and add blue crystals of sulfate in them, what happen? We observe that the whole water turns blue. This is due to the process of diffusion, because the blue color moves from its higher concentration to its lower concentration. Now, what is the significance? That it helps in the uniform distribution and helps in the exchange of gases. Also helps in attracting the pollinating agent, because the smells of flowers diffuses out in the environment, so it attracts the pollinating agents. And the second process is osmosis. Here, osmosis, it is the passive movement of water or any other solvent molecule from a region of higher water concentration to the region of lower water concentration through a semi permeable membrane. Here one thing is added, that is semi permeable membrane. Both are the same processes, but in osmosis there is involvement of the semi permeable membrane. Now, significance, it helps in the absorption of water from the soil. It helps in the opening and closing of stomata. It also helps in the growth of embryo during seed germination. Now, active processes. The active processes are uh, active transport, endocytosis, and exocytosis. First one is active transport. Active transport is the movement of ions or molecules mm, through a semi-permeable against the concentration gradient, and here energy sources are used. So energy, we obtained energy from the ATP molecule. Now, significance, it helps in the nerve impulse, muscle contraction, maintaining the balance, and the ultrafiltration of nephrons of a kidney. And now, endocytosis. Endocytosis, endo means inside the cell. Endo means in and cyto means cell. So endocytosis involves the intake of large size liquid or solid mm, macrobiomolecule inside cell through the flexible plasma membrane. Here we have example of amoeba. You can see the diagram of amoeba. This is the diagram of endocytosis in amoeba. Plasma membrane forms the pseudopodium and allow the entry of particulate material inside the cell. In the last diagram you can see that the plasma membrane closed out and the particulate enters in the cell, and this particulate is known as the phagosome. This is the process of endocytosis in amoeba. Endocytosis is of two types, um, pinocytosis and phagocytosis. Now what is pinocytosis? Pinocytosis is the process of intaking large size liquid biomolecules by the cells, which is called cell drinking or intake of micelles. Now second process is phagocytosis. Phagocytosis is the process of intaking of large size solid particle. Pinocytosis is cell drinking, liquid entry of liquid particles, and phagocytosis is the intaking of large size solid particles. It is also known as the cell eating. Now here exocytosis. Exo means outside, so it involves the expelling of large size material outside the cell through the plasma membrane. It is also called the cell vomiting. Vomiting means to expel out to expel out. So here cell is exocytosis is the cell vomiting. Example, expelling of residual body with undigested food, and release of secretion of glandular cells of liver, pancreas, etc. Here you can see the diagram of exocytosis. This is the diagram of exocytosis. This is the exocytotic vesicle, cell, secretion, plasma membrane, cytosol in first diagram. This rounded body wants to vomit, so it comes in contact with the plasma membrane, and in third diagram you can see that this cell, this exotic vesicle liberate out the undigested food or cell secretion released here. This is the diagram of exocytosis. This is all about the plasma membrane. Now the topic related with plasma membrane is cell wall. Cell wall is same as that of plasma membrane, but it is the outermost rigid freely permeable layer formed outside the plasma membrane. It is made up of different material. In plants it is formed of cellulose. In bacteria and blue-green alga cell it is formed of peptidoglycan. In fungi it is formed of chitin. Now what is the function? It is behaves as a protective and supportive coat for the cell. It also provides a definite shape to the cell. It works as a skin in works like a skin in human body. So it important function is protection and support the cell. Thank you.